Hi everyone, this is VKK Visalakshi from Biotechnology at KL University. Now we are going to learn about isolation of DNA from blood samples. What is DNA isolation? DNA isolation is a process of purification of DNA by using physical as well as chemical approaches. The first isolation of DNA was performed in 1869 by Friedrich Miescher. Techniques used to separate DNA are dependent on the source, age, and dimensions of the cell. Principle. Isolation of DNA usually begins with breakdown of cells. Lysate samples are mixed with phenol, chloroform, and isoamyl alcohol for separation of DNA and proteins. Proteins are denatured by the organic mixture. When the sample is centrifuged, the DNA will be retained in the aqueous layer. SDS will be present at the bottom of the tube and helps to denature the proteins and forms a cloudy interface. By adding alcohol to the mix, the DNA will be precipitated. The precipitated DNA is dissolved in the tris EDTA buffer, which can be stored for a longer time. Materials required: polypropylene, centrifuge tubes, Eppendorf tubes, micropipettes, gloves, centrifuge, B1 and B2 buffers, agarose, phenol, chloroform, isoamyl alcohol, ethanol, TAE buffer, and lastly, electrophoresis apparatus. Preparation of reagents. B1 buffer. Prepare 100 ml of buffer by adding 1.210 grams of tris HCl. pH should be 7.6, 0.744 grams of KCl, 2.032 grams of Mg, and 0.74 grams of EDT. Next, B2 buffer. Prepare 1000 ml of buffer by adding 0.121 grams of fresh HCl, 0.074 grams of KCl, 0.203 grams of MgCl, 0.074 grams of EDTA, 0.457 grams of NaCl. Tritonex. Prepare 0.1 ml of 100% Tritonex, 10% SDS. Prepare 10% SDS solution by adding 1 gram of SDS in the 10 ml of distilled water. Now lastly we should prepare 6 molar of NaCl. To prepare this, add 8.765 grams of NaCl in 25 ml of distilled water. Procedure. Collect 1.5 ml of citrated blood with the help of syringe into a vial. Add 900 ml of B1 buffer and 50 ml of Tritonex to the 300 ml of citrated blood in 1.5 ml vial and incubate at 37 degrees for 5 minutes. Centrifuge at 8000 rpm for 3 minutes and discard the supernatant. To the cell pellet, add 30 ml of B2 buffer and 40 ml of 10% SDS. Mix thoroughly and incubate at 37 degrees for 10 minutes. After incubation, add 100 ml of 6 molar NaCl and vertex to precipitate the protein. Centrifuge at 8000 rpm for 5 minutes and transfer the supernatant into a fresh tube and add 300 ml of isopropanol to it. Again centrifuge at 8000 rpm for 10 minutes to pellet down the DNA. Discard the supernatant and add 70% ethanol and spin at 8000 rpm for 5 minutes to remove any excess salt. Now discard the supernatant and air dry the pellet. Add 30 to 50 ml of TE buffer to dissolve the DNA. Now prepare 1% agarose gel and set the electrophoresis apparatus. Lastly, load 20 ml of DNA along with a tracking dye in the gel and electrophoresis at 50 to 100 volts. Stain the gel in ethidium bromide solution and visualize on the UV transilluminator. Result. We can observe orange color bands which represents the presence of DNA.